This video is going to talk about chromosomal abnormalities. As we've been talking about inheritance patterns, we need to make sure that we round out our conversation of chromosomal inheritance. We know at this point that chromosomes have implications in terms of linked genes, but they also have implications in terms of how chromosomes are distributed in meiosis, and then also if there's any excess processing before genes are passed on within gamete formation, and then they're ultimately expressed after fertilization. So there's two things here. There's abnormalities that can occur due to distribution and abnormalities that can occur due to processing or random mutations. So let's look at processing issues first. So we know that gametes are formed in meiosis, and this also is where chromosomes are distributed to those gametes. The main abnormality that you need to focus on here is non-disjunction, and this is the failure of chromosomes to separate evenly within this process. So this may result in one gamete having excess and one gamete having fewer than the normal number of chromosomes. If we look at this in terms of significance, we know that chromosomes are separated at two different times within meiosis. We have this first separation, which is meiosis 1, and then the second separation, which is meiosis 2. And the implications of when non-disjunction occurs can talk about the number of gametes that are lacking what they're supposed to. If we look at if we look at meiosis 1, we can see that there is non-disjunction for the separation of homologous pairs. Now, this is particularly important because you now have an entire new cell that is lacking a particular set of chromosomes, which means that the gametes formed from that cell will both be missing that chromosome. So what you end up with in terms of abnormalities in regard to non-disjunction at meiosis 1 is that you end up with two gametes with shortage and two gametes that have excess. And in all reality, you will have no normal gametes whatsoever. Now, this is just a little bit different when we talk about non-disjunction in meiosis 2. At this point in time, the homologous pairs have already separated. So we actually have two cells that have everything that they need. And what this means is the number of cells or the number of gametes that come out of this process, the number that have abnormalities will be fewer. Because if non-disjunction happens in just one of the cells, we'll see that there will be one gamete that is missing and one that has excess, but you see two that are normal. So we see one gamete with short, one gamete with excess, and then two that are normal. So it's not to downplay the significance of non-disjunction in meiosis 2, but the implications associated with non-disjunction in meiosis 1 are significantly greater because the number of gametes that have an abnormality at the end are significantly greater. And what that means is there's a greater likelihood of passing these abnormalities on to the gamete that is used for fertilization. All right, so there's some other things that you need to be aware of. And I apologize, my picture's covering up the slide. I'll try to move it here in a second. There's implications associated with what happens here. So aneuploidy results from the fertilization of gametes in which non-disjunction has occurred. And this is talking about specific gametes. A monosomic zygote means there's only one copy. That means that a normal gamete that is carrying a particular chromosome is fertilized with a normal gamete. And so what that means is it's carrying, one gamete is carrying nothing, and the other gamete is carrying its normal 1 version. So this has implications because we know that all of the traits will be expressed from that one gamete that is carrying 1 and no gene or no chromosome. We know that the version that exists is the one that will be expressed. And if that's a traditionally recessive gene, it'll be expressed regardless because there's nothing to cover it up. To the same regard, a trisomic zygote has 3 copies of a particular chromosome, and that is normally what results if the gamete from non-disjunction where both chromosomes went if that bonds with a normal one. So instead of having just, one chromosome from mom and one chromosome from dad, it's like getting one chromosome from mom and two from dad, or two chromosomes from mom and one from dad. But what that means is instead of having two chromosomes to deal with, you have three versions of that trait, which could complicate things horribly. Another word or vocabulary term associated with this is polyploidy, and this is a condition where you have extra chromosomes. So that's where these chromosomes from non-disjunction, these gametes that have extra, could cause particular problems. So if you have a gamete, or if you have a cell that has three versions of a trait, that's called triploidy, and if you have something that has four versions of a trait, it's called tetraploidy. And we know that these have implications just because more genes means or more versions of a gene only complicate two versions. We now have four, which means our probability of being able to predict outcomes is significantly reduced. All right, so those are issues associated with distribution issues. So we can also talk about these ideas of chromosomal modification or chromosomal manipulation. So this is where we're talking about reorganization of parts of the chromosome. So this has no implication on meiosis whatsoever. This has implications on random mutations of the structure of the chromosome. We could have a deletion where part of the chromosome is lost. We could have a duplication where part of the chromosome is repeated, an inversion where part of the chromosome is removed, flipped, and then reinserted. Or we can have this idea of a translocation where part of a chromosome is removed and then replaced on a different chromosome. So you end up with one chromosome that doesn't have everything that needs, and then another chromosome that has more than it needs. And we can look at what this looks like here in a second. So if we talk about a deletion, this is where part of a chromosome is removed and then never re-added or never reinserted. So in this case we have this letter D, or gene D, and what happens there is that is removed, and then the chromosome realistically slides together as if it was never there. And that's particularly important because if we are looking for the determination of one gene, if we have two genes and one of them is missing, it doesn't matter what the other version is, that's the one that's going to be expressed. To the same regard, duplication causes some issues because if we have this section, this B and C section, a duplication means it's not only in its normal position, it is also duplicated in a different position. And so this almost acts like a triploidy situation, that this chromosome is donating two versions of these traits, whereas it's only expected to bring one. So you not only have these two versions from this chromosome, you have the other version from the other donated gamete, which replicates this triploidy-like scenario. The two others are an inversion. Inversions are where you have the same genes, but they're in a different order. And this may actually not have huge implications in the event that the genes or the part of the chromosome contains a certain amount of genes. The order in which they're read might be different, or if this is a part of a larger gene, there would be significant implications. But due to the fact that there's not a loss or a duplication, sometimes it's really difficult to predict which way it's going to go, whether it's going to be drastic or whether it's going to be insignificant. But the idea here is that this is removed and then it is flipped 180 degrees and then reinserted. A translocation can be particularly problematic as a part of one chromosome is removed and then added to a different part of that chromosome or a different chromosome completely. It does not have to be an exchange like is depicted here in the particular picture. Sometimes it can even be A and B is removed from here and then tacked onto this chromosome so that you end up with A, B, M, N, O, P, Q, R. And part of this chromosome is never removed. So a translocation is just removal of one segment and it being tacked onto another. All right, now on Wednesday, we're going to look at the significance or genetic diseases associated with each of these implications.